Welcome to this GCSE Biology revision guide on enzymes. We will explore everything you need to know about biological catalysts, how they work, and why they are vital in your body and beyond. This video will cover 4 main areas: what enzymes are, how they work, factors affecting their activity, and their role in digestion. Let's start with Chapter 1, what are enzymes? We will establish the fundamental nature of biological catalysts. Enzymes are biological catalysts. A catalyst speeds up a chemical reaction without being permanently changed. Enzymes do this inside living organisms, making life-sustaining reactions possible at body temperature, unlike industrial catalysts requiring extreme heat. Crucially, enzymes are made of proteins, which are long chains of amino acids folded into a precise 3-dimensional shape. This specific shape is essential for their function, but also makes them vulnerable to changes in their environment. Every enzyme has a highly specific, unique 3D shape determined by its amino acid chain. This shape dictates the chemical reaction the enzyme can catalyze. If the shape is lost, the function is lost. The most important feature is the active site, where the substrate binds to form an enzyme-substrate complex. The active site's unique shape is complementary to only the correct substrate. The reaction occurs, products are released, and the enzyme is ready to be reused. Chapter 2 focuses on how enzymes work, covering the lock and key model, substrates, and enzyme-substrate complexes. The lock and key model is the key to understanding how enzymes work. A specific substrate fits only one enzyme's active site, just as a key fits only one lock. This explains the high selectivity of enzymes. The process involves 4 steps. The free enzyme is available. The substrate binds precisely to the active site. The enzyme catalyzes the reaction, forming product molecules. And finally, the products are released, leaving the enzyme unchanged and ready to act again. A key exam point is that the enzyme is not consumed or permanently altered in the reaction, allowing a single enzyme molecule to catalyze the same reaction thousands of times per second. This reusability depends on the enzyme maintaining its correct 3D shape. This is a frequently tested exam idea. Shape determines function. Remember 3 scenarios: correct shape for efficient catalysis, changed shape leading to ineffectiveness, or the wrong substrate which prevents binding and reaction. Chapter 3 covers the factors affecting enzymes, including temperature and pH, and what happens when these conditions change. As temperature rises, molecules gain kinetic energy, increasing collisions and the reaction rate up to a point. Every enzyme has an optimum temperature, approximately 37 degrees Celsius for human enzymes. Temperatures above this cause denaturation. When the temperature rises significantly above the optimum, the bonds holding the protein's 3D structure break. This process is called denaturation. The active site changes shape permanently, destroying the enzyme's ability to function and preventing recovery. Denaturation is the permanent change in an enzyme's 3D shape caused by excessive heat or extreme pH. When the bonds maintaining the shape break, the active site changes, and the enzyme can no longer bind to the substrate. This temperature graph is classic exam material. The rate starts low, rises steadily to the optimum at 37 degrees Celsius, and then drops rapidly to 0 as denaturation occurs. pH measures acidity or alkalinity. Changes in pH alter the charges on the amino acids, disrupting the bonds that maintain the enzyme's shape. This changes the active site, reducing or stopping activity. Every enzyme has an optimum pH. Pepsin, found in the highly acidic stomach, has an optimum pH of about 2. Salivary amylase in the mouth works best at neutral pH 7, while pancreatic enzymes in the small intestine prefer a slightly alkaline pH of 7.5 to 8. Similar to heat, a pH too far from the optimum causes the enzyme to denature. Hydrogen ions disrupt the internal bonds, permanently altering the active site's shape. This irreversible change means the substrate cannot fit, and the reaction stops. Both temperature and pH follow the same general principle. Both have an optimum where the reaction rate is maximum, and moving too far in either direction leads to denaturation, where the active site changes shape. We now move to Chapter 4, Enzymes in Digestion. We'll explore how your body uses enzymes to break down food into molecules it can absorb. Digestion requires enzymes because the complex food molecules we eat, starch, proteins, and fats, are too large to pass through the intestinal wall. Digestive enzymes break them down into smaller, soluble molecules like glucose, amino acids, fatty acids, and glycerol for absorption. The 3 key digestive enzymes are amylase, protease, and lipase. Each is specific to a different type of large food molecule. Amylase breaks down starch into maltose, which is further broken down into absorbable glucose. It is produced in the salivary glands and pancreas, acting in the mouth and the small intestine. Proteases break down proteins into amino acids. They are produced in the stomach, pancreas, and small intestine. Pepsin in the stomach is a key example, adapted to the low pH of gastric juice. Lipase breaks down fats into fatty acids and glycerol. Bile, produced by the liver, emulsifies fats, increasing the surface area for lipase to work efficiently. Remember, bile is not an enzyme. It doesn't catalyze a reaction. This diagram shows the main organs involved in digestion. The pancreas is particularly vital, producing all three major digestive enzymes: amylase, protease, and lipase. Here's a summary table for the three main digestive enzymes, covering their substrates, products, production sites, and where they act in the body. We have reached the end of the video. Let's quickly review the key points to remember. First, enzymes are biological catalysts, speeding up reactions without being permanently changed. Second, they are proteins, and their 3D shape, particularly the active site, is essential for function. Remember to use the term denatured when explaining the effect of high temperature or extreme pH, and never say an enzyme is killed. Enzymes are unchanged and reusable after a reaction, and bile is not an enzyme. Make sure you know the key vocabulary. A catalyst speeds up a reaction. The active site is where the substrate binds. Denaturation is the permanent shape change due to heat or pH, and the optimum is the condition for the fastest rate. Test your knowledge with these practice questions. Pause the video now to try answering them without your notes first. Think about the functions of catalysts and the details of the lock and key model. You've got this. Enzymes are a fundamental topic in biology. Remember that shape determines function. Review, recall what you've learned, and apply your knowledge to practice questions. Good luck with your revision.